Let's just dive straight into this explainer. Welcome to a literal cinematic journey inside your own cells. Today we're taking dense textbook biology and we're going to transform it into a microscopic blockbuster movie. We're looking at the ultimate blueprint of life itself, DNA replication.
Now, the vision behind this is just so cool. I want you to imagine this prompt, a cinematic, hyper-realistic 3D medical animation. A glowing translucent DNA double helix is being unzipped by a rotating helicase enzyme. That's not just a fancy sentence, right? That is the actual starting point to visualize the incredible, high-tech documentary style we're exploring today. It totally sets the stage for a mind-blowing biological process.
Here's our director's call sheet for today. First, entering the cell nucleus. Then, unzipping the blueprint. Next, the leading strand, followed by the lagging strand twist. And finally, the final masterpiece.
All right, part 1. Entering the cell nucleus. So picture this. Deep inside the nucleus of your cells, there is a masterpiece of biological engineering happening right this very second. Imagine drifting through this dense, fluid environment. It's lit up with cinematic, bioluminescent blue and purple lighting. Floating all around are bits of chromatin, and drifting gracefully through this heavy depth of field is a perfectly twisted DNA double helix. It's quiet, it's totally ambient, right before the massive machinery kicks into gear. And this actually grounds our whole cinematic scene in real, hard science. You see, this process doesn't just start randomly anywhere on the DNA. There's a very specific region where the replication originates, which, appropriately enough, is called the origin of replication. But here's the catch. Physically pulling apart these long, intertwined strands of DNA, that takes a massive amount of energy. It's a hugely expensive energetic process for the cell to run.
So, how exactly does the cell foot that massive energy bill? Well, enter DNTPs, or deoxyribonucleoside triphosphates. These brilliant little molecules are ultimate multitaskers. Not only do they act as the physical substrates, literally the physical building blocks for the new DNA, but their terminal phosphates actually provide the high energy required for the reaction to happen, kind of like ATP does. It's like the cell is building a house using glowing puzzle pieces that actually pack their own internal batteries. How cool is that?
Now let's meet the absolute stars of our microscopic high-speed production. Our core cast of biological machines includes helicase, which is basically our heavy-duty motor, DNA polymerase, the massive, complex master builder, and ligase, which acts as the vital glue. These 3 enzymes are going to drive the entire plot of our explainer today.
Moving into Section 2, Unzipping the Blueprint, and Action. Okay. Before the heavy machinery goes into overdrive, let's look at the logical flow of our plot. We've got 3 core phases here. Phase 1 is the initial unzipping. Phase 2 is building the leading strand. And Phase 3 tackles the much trickier lagging strand.
Let's hit Phase 1. Enter helicase. Imagine this wedge-shaped, microscopic, incredibly high-speed motor emitting this soft, bioluminescent golden glow. It locks directly onto the twisted double helix and just powers forward, physically unwinding and ripping those intertwined genetic strands apart. It is forceful, it is insanely precise, and it kicks off this whole chain of events.
Now, as helicase unzips those strands, it creates what we call a replication fork. You notice that Y shape? Well, because separating a massive DNA molecule along its entire length would just completely bankrupt the cell's energy budget, it only opens a tiny section at a time. This replication fork is our localized, highly active movie set where all the subsequent action goes down.
Which brings us to Section 3, the leading strand. With the DNA unzipped and ready to go, our next star character makes a dramatic entrance into the replication fork. Meet DNA polymerase. This thing is a massive, incredibly complex biological machine. It is the absolute master builder. It smoothly clamps right onto one of those newly separated genetic strands, totally ready to get down to the crucial work of copying the code. On this leading strand, DNA polymerase performs what we call a continuous build. It just glides swiftly forward in one fluid, uninterrupted motion. And surrounding it, you have thousands of free-floating nucleotides just swarming around like glowing dust motes in the cellular fluid. The polymerase grabs these puzzle piece molecules and perfectly pairs them up, locking them into the exposed DNA to form the new double helix. I mean, it is a highly satisfying, totally nonstop assembly line.
But then we hit Section 4, the lagging strand twist.
Okay, so the leading strand is smooth sailing. But how on earth do you build backward? This right here is a massive biochemical plot twist. The other half of the unzipped DNA runs in the exact opposite direction. And our biological machines, well, they have some very strict directional rules they absolutely have to follow. Here is the major complication. DNA polymerases can only catalyze in one single direction, specifically from the 5' to the 3' direction. It is a strict one-way street. Our master builder is physically incapable of just throwing it in reverse and smoothly backing up the other side of the fork.
So unlike that beautiful fluid motion of the leading strand we just saw, the lagging strand goes through discontinuous synthesis. Because the polymerase can only build one way, it's forced to work in these short, frantic, jumping bursts. It builds backward a bit, detaches, jumps forward toward the fork, and then builds backward again. These newly built, segmented, glowing chunks even have a name: Okazaki fragments. It is a crazy, fragmented way to build. But hey, it works.
But now we're left with all these disjointed chunks on the lagging strand, right? That is exactly where our third crucial character comes in: ligase. Ligase is absolutely the unsung hero of this whole operation. It rapidly swoops in to act as the glue. It flawlessly stitches those discontinuous, glowing Okazaki fragments together into one unified, continuous genetic strand, perfectly matching the perfection of the leading side.
And that brings us to Section 5, the final masterpiece.
Let's pan the camera all the way out now and look at the final cut of this incredible process. What started as one single original DNA double helix has now successfully split and mirrored itself completely. In just moments, we are left with two identical, flawlessly twisted DNA double helices just drifting gracefully together in that deep blue cellular fluid.
Millions. That is exactly how many times this flawless, microscopic miracle is happening inside your own body, inside your own cells, every single day. The speed and the sheer accuracy of it are just absolutely staggering to think about. Which brings us to our final thought. If copying our genetic blueprint requires such astonishing speed, high-energy budgets, and absolute molecular precision, is human biology the greatest feat of engineering ever directed? Really think about that the next time you marvel at a cinematic masterpiece.